My name is Satvir Yadav. I am pursuing my master's degree in food technology from Banaras Hindu University. I am the student of the first year food technology MTech. Now we are going to discuss nutrigenomics and the differences between the nutrigenomics and nutrigenetics, and then we will discuss about the single nucleotide polymorphism, and then nutrition gene interaction, and what are the advantages and the weaknesses of the nutrigenomics. And we will discuss about the conclusion and what are the future challenges in the field of the food science technology in context of the nutrigenomics. So now what we are going to discuss, what is nutrigenomics. So there are the so many nutrient incorporated in the human body, and the what are the interaction between the nutrient and a particular genome. And we know that the genome is the genome is the made by the complex of the DNA, and we will discuss about the interaction between genome and the nutrient. तुम देखो इसको. So basically, we are going to discuss the study of the consequences of the influences of the nutrient and the other bioactive compounds that are the present in the food on the expression of the genetic material. And we know the nutrigenomics is a branch of the nutritional genomics and is the study of the effect of the food and the food constituent on the gene expression. We know that there are the so many diseases that are the that are the that are the there are the responsible for the diseases so many nutrient and a particular genes, and there are the so many interactions between the genes and the particular nutrient, and we are going to discuss how the particular genes and the particular genome is the react with the nutrient, and the what kind of the disease and the complex situation is the created in the human body. And it will also determine the individual nutrition requirement based on the genetic makeup of the person as well as association between the diet and the chronic disease. It will identify the gene involved in the physiological responsible to diet and the gene in which small changes called the polymorphism, and the influence of the environmental factor on the gene expression. So basically the how the environmental factors and the nutrient is react with the genes, and there are the so many changes in the human genes are made by the particular food material, and there are so many bioactive compounds that incorporated in the food. So basically there are the two terms, nutrigenomics and the nutrigenetics. Both are the just like a two side of a coin, and how the food nutrient and the genome interact with the food, both term can be explained. So what is the nutrigenomics? The application of the genomics in nutritional research, like the example, the way in which the food ingredient influence the gene, and the nutrigenetics, study of the individual differences at the genetic level influences genetic response. So these two terms basically discuss in the context of the nutrigenomics. So there is a very small difference between the nutrigenetics and the nutrigenomics. So nutrigenetics, like your genes influence how your body react to the food, and nutrigenomics, what you eat influence how your genes behave. That is the basic difference between nutrigenetics and nutrigenomics. So there are the four basic tenets of the nutrigenomics. First is improper diet, a risk factor for the disease, like the particular disease is the responsible for an improper diet. And second is the dietary chemical, how alter gene expression or change genome structure. And third one is the degree to which diet influences the balance between healthy and the disease state may depend on an individual genetic makeup. And fourth one is Some diet regulates genes are likely to play a role in the onset, incidences, progression, and severity of the chronic disease. And next is nutrigenomics and the nutrigenetics deal with few concepts. Specific dietary profile can modulate the delicate balance between health and the disease, acting either directly or indirectly on gene expression. And the individual genetic makeup, that is the presence of the polymorphism in nutrient regulate genes, affects individual risk of the disease. So basically the meaning of this concept is that a particular genes is the responsible for the disease, and how a particular makeup of the genome is responsible to cure and to fight a disease. So this is the basic science of the nutrigenomics. How does diet affect the gene expression? So gene express themselves through protein, enzyme are the special proteins designed to get things started. Our genome instruct ribosomes to produce many enzymes that destroy toxins. So basically our ribosomes is the responsible to release many enzymes and some other biochemical compound, and these compound found in our body and fight with that. There are the different chemical available in our body. So these activity act on our genetic materials, and these activities are regulated by the different type of the nutrient. Next topic is the single nucleotide polymorphism. So, in the single nucleotide polymorphism, a specific gene polymorphism in human population change their metabolic activity. After the changing the metabolic response, a particular diet that are the influence a particular pattern of the disease, and some SNP change the recipe. After the changing the recipe, a particular protein is produced, or structure of the particular protein may be altered. After the altering the particular protein structure, that may be responsible for a particular disease. Next, we are going to discuss nutrition and gene interaction. So there are the basic type of the nutrition and gene interaction. First is the direct interactions. So in the direct interaction, nutrient after interacting with the receptors behave as the transcription factor that can bind to DNA and induce gene expression. Second is epigenetic interactions. In epigenetic interactions, nutrient can alter structure of the DNA so that gene expression is altered. And third one is genetic variation. In genetic variation, common genetic variations such as the single nucleotide polymorphism can alter the expression or functionality of the gene. Next is gene disease interaction. Nutrigenetic disease, 97% of the genes have known to be associated with the human disease result in monogenetic disease. Modifying the dietary intake can prevent some monogenetic disease. Example, phenylketonuria food containing the amino acid phenylalanine, including high protein foods such as fish, chicken, egg, milk, cheese, and dried beans. And these kind of the food can prevent some kind of the disease. And in case of the defective aldehyde dehydrogenase enzyme, alcohol must be avoided. So these are the some kind of the nutrient can be prevent for the disease, and some kind of the nutrient that are not digestible in presence of the particular compound. So these gene and diet disease interaction can be very useful in context of the curing many disease and preventing so many disease. So there is a study, nutrigenomics and the obesity, a study based on the contribution of the genes to body weight in lipoprotein response. So in this studies, 28 pairs of the monozygotic twins are a high carbohydrate diet for the 6 weeks, then they switch to a high fat. So in these study, how the particular gene is the responsible for the obesity and the particular workout is the perform in the study, and the how obesity intake and the and the low density lipoprotein and the high density lipoprotein is the very a particular pair of the monozygotic twins is the uh, studied in the particular case. So now we are going to discuss about the advantage and the disadvantage of the nutragenomics. So there are the some advantage of the nutragenomics, like the it increase the focus on a healthy diet and the lifestyle, and we know that in the recent day how the healthy diet can be maintained in the food system. This is the very big challenge, and this is the very useful and a very recent advanced topic in the field of the nutrition. And second advantage is the focus on the prevention of the disease. How can we cure a disease by the incorporating the food nutrient and the food component in the food? And so this is the very useful in branch of the food science technology. And there are the some weaknesses of the nutrigenomics. It especially focus on a particular nutrient, and there are the some misleading claim available in the some research. So this is also a weakness and the drawbacks of the nutrigenomics. And now we are going to discuss some conclusion and the what are the future challenges in the field of the nutragenetic, and we know that the food safety and the health can't be ignored in the field of the food technology and the food processing sector. And we know that there are the some nutrient and some uh, some component that are the used by the food technologist, and these all the nutrient are the responsible for the so many factors, and how these nutrient can be incorporated and can be useful and can be beneficial for our the body, and so that a particular safety of the food and the health can be maintained by the field of the nutrigenomics. And nutrigenomics must be utilized in addressing the growing epidemic problem of both under and overnutrition, and some nutrition problem like the overnutrition and undernutrition. So and there are the many challenges over the nutrition can be cured by the help of the nutrigenomics. And thank you so much for listening all the things regarding the nutrigenomics.